So I have the pleasure of introducing our next speaker today, which is Dr. Joan Conaway. I guess I was drafted to introduce Joan for various reasons. So even though I don't work on transcription anymore, so I have to tell Joan this story. Actually, so when I was a student, I was actually working on transcription. So the very first time I saw you from a distance was actually in Heidelberg 20 years ago at a transcription meeting. So I think that you made a lasting impression because I think you were one of the better prepared speaker at the conference. And little did I know that actually many years later I became Joan's colleague at the Stowers Institute before I moved back to HKUST here. So, and another personal kind of lesson that I learned from interacting with Joan over the years is actually it's okay to do great science and it's okay to play nice in science. And so you win, and I think the reputation is actually really coming from kind of the personal quality as well as scientific quality. So I won't kind of take up too much more of your time. So welcome very much, Joan, for giving us a lecture.
Has been to try to contribute to an understanding of the mechanism and regulation of transcription by Pol II. As you heard this morning from Bob Rader, messenger RNA synthesis is really a very complex process that involves the interplay of a huge number of transcriptional regulatory proteins, and I won't try to cover quite as many of them as Bob did this morning, but suffice it to say that there's a lot of stuff going on in the regulation of transcription. Now, as you know, the proper execution of the transcription program is really critical for normal growth and development of all organisms. And the misregulation of that program is at the heart of all kinds of disease states, ranging from developmental defects to cancers. And so over the past several decades, a major goal of many labs, including ours and those of a number of the people who are in the room today who have been here in Hong Kong for this meeting, has been to try to identify the network of regulatory proteins that control transcription by Pol II, and then to unravel their mechanisms of action. So in my talk today, I'm going to focus on work in the lab that seeks to understand how a major regulatory complex that Bob Rader introduced to us this morning contributes to transcriptional regulation. Now, before I go on, I want to acknowledge the colleagues and collaborators who are responsible for the work that I'm going to describe. First of all, Joan Conaway, who's here in the audience and with whom I co-direct the lab. Current lab members, Amal Ranjan, Cherry Tomomori Sato, Shigeo Sato, and Yanfeng He. Amal has been primarily responsible for the genomic work that I'm going to describe today, with a great deal of collaborative help from Cynthia Chen in the Stowers Bioinformatics group. I'd also like to acknowledge past lab members, especially Hide Takahashi, who's now a professor at Yokohama City University. Hide initiated a lot of the key parts of the story that I'm going to describe today. And we've continued to collaborate extensively with him since he went back to Japan to set up his lab. Then I'd also like to make special mention of Mike Washburn and Laurence Florenz in the Stowers Proteomics group, with whom we've collaborated extensively over the years. Okay, so as you heard this morning, RNA polymerase II is sort of a stupid enzyme. It can't initiate transcription on its own. And we and Bob Rader and Jean-Marc Egli and others in the field showed quite a few years ago now that RNA polymerase II needs help from a set of general transcription factors called TF2D, TF2B, TF2E, TF2F, and TF2H, simply to find a promoter on DNA, to unwind the DNA template around the transcription start site, and to initiate transcription. Now, although it's not required for promoter-specific transcription in the test tube, the mediator complex, this very large multi-subunit complex, can bind tightly to RNA polymerase II and the initiation factors at the promoter, at the promoters of protein-coding genes, as well as genes that direct the synthesis of various non-coding RNAs, as I'll describe today. Now, based on the observation that, like Pol2 mutations in core mediator subunits lead to a complete block to transcription in yeast, mediators sometimes been considered to be an additional component of the general transcription apparatus, like RNA polymerase and the general transcription factors. However, I should say that mediator, either in whole or in part, appears to be substantially less abundant in some terminally differentiated cells, suggesting that at least the intact mediator may be dispensable under some conditions. So whether it's truly a part of this general transcription apparatus sort of remains to be determined. Now, of note, and Bob alluded to this morning, mutation or overexpression of quite a number of the mediator subunits is associated with a whole range of developmental diseases and in cancers. And in at least some cases, those mutation or overexpression have been shown to be causal. And so understanding how the mediator contributes to transcriptional regulation is, in the long run, potentially of clinical importance. All right, so the detailed mechanisms by which the mediator contributes to transcriptional regulation, despite quite a few years of study, are still not very well understood. Hints came from the fact that the mediator was first identified back in the early 1990s by Roger Kornberg and his colleagues, who purified it from yeast as an activity that was needed to reconstitute activator-dependent transcription in a purified transcription system consisting of Pol2 and these general transcription factors. And it's clear that different subunits of the mediator complex can bind directly to different DNA binding transcription factors, as you heard from Bob this morning. And as a consequence of these interactions, these mediators, the mediator can transmit both positively acting and negatively acting signals from these DNA binding transcription factors through to RNA polymerase II. Now, in large part because of its ability to drive activator-dependent transcription or recruitment of Pol2 and general factors to promoters, both in vitro and in vivo, most attention in the early years focused on the idea that the mediator acts primarily to control initiation of transcription. But of course, the story is a good deal more complicated, and as I'll describe, there's now evidence that mediator has important roles in other stages of transcription as well. So once polymerase has initiated transcription, of course, it has to move away from the promoter to form the elongation complex and ultimately to complete synthesis of the nascent transcript, to release that transcript, and to terminate transcription. And it's become increasingly clear that transcript elongation, just like transcription initiation, is a highly regulated process. For example, the expression of a very large number of genes is controlled by a mechanism that involves a process called promoter proximal pausing, in which polymerase initiates transcription but moves just a short distance from the promoter, where it then stops and sits until some regulated event causes it to be reactivated and converted into a productive elongation complex. The details of the molecular mechanisms, again, regulating that process are still being worked out, but there's good evidence that the pause is induced in part by the combined action of negatively acting transcription elongation factors called SPT4 and 5, and NELF, which stands for negative elongation factor, while reactivation of paused RNA polymerase depends on a host of elongation regulators. And the ones that are relevant to this talk today are a factor called PTEF-B and factors called ELL and EAF, which form a heterodimer that can control, and working together, these guys can help to reactivate promoter proximally paused polymerase. And I should say that while the best characterized functions of these guys are in the early stages of transcript elongation, in the relief of this promoter proximal pausing step, they likely also contribute to elongation control later, perhaps throughout elongation of the entire transcript. So today what I'm going to do is argue that in addition to these sorts of elongation factors, the mediator complex also plays key roles in the control of post-initiation events, including both transcript elongation, and as I'll describe in recent results, we have obtained evidence that the mediator complex can even influence steps at the far end of the transcription unit. At some genes, it can contribute to the regulation of transcription termination and of 3' end processing to form mature RNAs. And in particular, what I'm going to do is describe evidence that a mediator subunit that's called MED26 functions genome-wide to regulate gene expression through a mechanism that involves the recruitment of at least 2 kinds of complexes that contain these 2 proteins, ELL and EAF. One of them was dubbed, not by us, the super elongation complex, or SEC, and the other was dubbed the little elongation complex, or LEC. Now, before I go on to tell you about that work, I want to describe just a little bit more about the mediator complex. So, since its initial identification, studies from the Kornberg lab, the Rader lab, our lab, and a number of others define the mediator complex as a very large complex that's composed of 25 subunits in yeast and even more in metazoan cells, including vertebrates and humans. So the mediator has been shown to consist of a core complex composed of 3 subassemblies referred to as the head, the middle, and the tail modules. And as Bob alluded to this morning, the tail module is a target for interaction by many of the DNA binding transcription factors, as are subunits and surfaces near the tail module in this region. And I should mention that this beautiful image that I'm showing you is a low resolution EM reconstruction done by Francisco Asturias of some of the human mediator preparations generated in our lab. Now, the additional submodules, the head and the middle, provide surfaces that bind to RNA polymerase II, and by mechanisms we don't understand, again, help to transmit signals from activators interacting with the tail and other surfaces through to RNA polymerase II. Now, a portion of mediator also contains a so-called kinase complex that's dissociable. It's found on some mediator, but not on all mediator. It consists of a kinase cyclin pair, CDK8 and cyclin C, and these 2 subunits, MED12 and MED13. And the kinase module binds to the core in a way that occludes the assembly of RNA polymerase II with the mediator complex. And for that, and in part for that reason, for a long time, the kinase module was thought to have largely a repressive function in mediator activity. However, I should say that early genetic studies in yeast, as well as more recent work from Joaquin Espinoza and others, argue that in some contexts, paradoxically, this kinase complex, despite the fact that it prevents the interaction of the mediator core with RNA polymerase II, is required for activation of transcription. Okay, a second form of mediator includes this subunit MED26, and that's specifically in metazoan cells. MED26 orthologs are not detectably found in any fungi, so this carries out functions that have been acquired later in evolution. Now, although one can isolate from cells a little bit of mediator that contains both MED26 and the kinase module, the vast majority of mediator that has MED26 lacks the kinase. And several lines of evidence suggest that MED26 has a particularly important role in activating functions of the mediator complex. First of all, mediator that contains MED26, if you're careful during isolation of the mediator complex, pulls out near stoichiometric amounts of RNA polymerase II. And so it interacts, mediator containing MED26 interacts with the target for regulation. Second of all, work from a number of labs, including the Tijen lab, the Meister-Ernst lab, and others have shown that in reconstituted reactions like the ones that Bob showed you this morning, MED26 containing mediator acts as a co-regulator for activator-dependent transcription in vitro. And finally, work from Achim Lutz's lab showed for several genes that activation in cells appeared to be dependent specifically on a population of mediator that's associated with MED26. And so for all of these reasons, about a decade ago, we became particularly interested in addressing the question why MED26 might contribute to the activating function of mediator. By what mechanism might it help mediator to support activation? And so to begin to address that question, we performed a large proteomics screen to look for mediator interacting proteins. And basically what we did was generate cell lines that expressed epitope-tagged MED26, as well as epitope-tagged versions of all of the other mediator subunits. And then we purified through flag immunoprecipitation mediator and associated proteins, and working closely with our proteomics colleagues, then went ahead to identify proteins that co-purified with the mediator complex. What we found was that mediator purified through all subunits except for MED26, basically co-purified with mediator, with small amounts, very small amounts of other proteins, some of which were interesting. But we found that when we purified mediator through a flag epitope tagged version of MED26, the situation was quite different. So mediator purified through MED26 consistently contained the Pol2 elongation factors that I told you about earlier, ELL, EAF, and PTEF-B. It also contained a couple of then unknown proteins, which are now referred to as ICE1 and ICE2 for interacts with the C-terminus of ELL, as well as a collection of interesting proteins that had been identified as translocation partners of this histone methyltransferase in various leukemias. Now, after a series of structure-function experiments, Cherry and Shigeo Sato and Terry Parmalee in the lab were able to show that a conserved domain in the carboxy terminus of MED26 and its CTD was sufficient for binding to mediator that in turn binds to RNA polymerase II. And they showed further that this N-terminal domain, or NTD, as I will probably refer to it a lot during the talk, was sufficient and necessary for binding to all of these other proteins. Now, based on a variety of of biochemical experiments that I won't drag you through, Hide Takahashi, while he was in the lab, went on to show that the MED26 N-terminal domain binds to at least these two types of ELL and EAF containing complexes, the super elongation complex and the little elongation complex that I alluded to before, and that the MED26 N-terminal domain binds to both of these complexes through direct interactions with this protein called EAF. Okay, so what are these things anyway? Well, The first of these MED26 binding complexes was identified about the same time by a number of labs, including ours, Ali Shalatafard's, Monsef Benkaran's, Cheng Zhao's, and later others. And it came to be called the superelongation complex because it contains two kinds of elongation factors, PTEF-B and ELL and EAF, that were known to control RNA polymerase II by completely different mechanisms. So PTEF-B was first identified in David Price's lab. As I told you earlier, it regulates the transition of promoter proximally paused Pol2 into productive elongation complexes. It's a kinase-cyclin pair that consists of the cyclin-dependent kinase CDK9 and cyclin T. And what it does is to phosphorylate SPT4 and 5, which, as I told you, acts as a negative regulator that helps to promote the promoter proximal pause. And it phosphorylates the other negative acting elongation factor, NELF. And when it phosphorylates these proteins, SPT4 and 5 is converted from a negative factor to a positive factor, and NELF is kicked out of the elongation complex. PTEF-B also phosphorylates RPB1, and RPB1 is the largest subunit of RNA polymerase II. It has an unusual carboxy terminal domain, or CTD, that's composed of many repeats of the heptapeptide sequence YSPT-SPS. In humans, repeated 52 times. And PTEF-B phosphorylates the RPB1 CTD on this residue, serine 2, of its heptapeptide repeats. And in vitro can also phosphorylate this serine 5 position. Okay, now the second elongation factor in this complex acts by a phosphorylation-independent mechanism. It's composed of members of the ELL and EAF family of proteins, and what it does is to interact directly with elongating RNA polymerase II and stimulate the overall rate of transcript elongation. And I want to just show you what exactly I mean by that. So by saying that it stimulates the overall rate of elongation. This is an assay from Ali Shalatafard's original characterization of the ELO protein and its ability to stimulate elongation by RNA polymerase II. So what Ali's done in this experiment is to assemble pre-initiation complexes at a promoter with purified, or actually this is a different assay. This is not a promoter-specific assay. What Ali's done here is to initiate, using a trick, RNA polymerase on a double-stranded piece of DNA and allow it or cause it to synthesize these transcripts of 135 nucleotides in the presence of low concentrations of radiolabeled CTP as well as ATP and GTP. And this template has a so-called ULIS cassette that allows it, in the presence of those nucleotides, to make this 135 nucleotide transcript. And then what he does is add to these reactions UTP to allow polymerase to escape from this 135 nucleotide position, as well as high CTP to dilute the label. And so you don't see any new initiations, you just watch these transcripts get longer. And what you can see here is that in the absence of ELL, under the conditions of these reactions, RNA polymerase II rather slowly elongates, generating longer and longer transcripts until it reaches this position at the end of the reaction. But in the presence of ELL, the polymerase quite quickly moves from this position into longer transcripts. And by the end of the reaction, a very large fraction of the enzymes have made it to pretty close to the end of the template. Now, what about the EAF proteins? Well, EAF1 and EAF2 were both identified as ELL-associated factors in yeast 2 hybrid assays by Michael Thurman and his colleagues. And Stephanie Kong and Charles Banks in the lab showed that the EAF and EAF EAF1 or EAF2 proteins through their ability to interact with ELL would further enhance ELL's ability to stimulate elongation. And this time we are looking at promoter-specific assays where Charles and Stephanie have incubated Pol2 and the general factors with a promoter that contains, or with a template that contains a promoter that in the presence of 3 nucleotides allows synthesis of these short transcripts. And these can then be chased into longer transcripts again by adding more nucleotides. And in the first lane here, they've not added any additional elongation factors. And you can see that the polymerases are sort of stalling out in this region. If they add EAF protein by itself, again, the polymerases stall out. But if they add both ELL and EAF if they add ELL by itself, they see some stimulation, but when they add both, a large fraction of the polymerases in this reaction make it, again, to the end of the template. So, again, ELL and EAF acting together interact directly with elongating Pol2, and they make it transcribe more rapidly. So the other thing I wanted to mention is that the ELL gene had actually first been identified in Janet Rowley's and Harai's labs as a translocation partner in acute myeloid leukemia of the MLL gene, as I mentioned. The MLL gene is a histone H3K4 methyltransferase. And what happens in this translocation is that the N-terminal portion of MLL, which lacks the catalytic domain but contains domains required for targeting MLL to target regions in chromatin, is translocated to almost the entire ELL protein, including all the regions that are required to stimulate elongation by RNA polymerase II. And so what that does is generate a new protein that contains targeting sequences that can mistarget ELL to the wrong places in chromatin at the wrong time. Of note, these additional proteins, AFF1 or AFF4 and ENL or AF9, are also found as translocation partners of MLL in leukemias. And it turns out that MLL, ELL, as well as these other MLL fusion proteins, all are able to assemble into complexes like these. And so a popular model in the field is that these MLL chimeras can mistarget the superelongation complex, thereby giving rise to the cancers. So what does this thing do in cells? Well, PTEP-B, ELL, EAF, and other components of the SEC have been shown to work together to regulate the expression of a whole bunch of different protein coding genes, including oncogenes like these MYC and FOS, stress response genes, developmental regulators, viral transcripts like the HIV LTR, and many, many, many other protein coding genes. The second ELL-EAF containing complex that's bound by MED26, the little elongation complex, contains ICE1 and ICE2, as well as this additional protein, a zinc finger protein called ZC3H8. There's a lot less known about the little elongation complex, but like the SEC, it appears to be capable of, it appears to be a positive regulator of transcription. HEDA has shown that it can stimulate Pol2 elongation in vitro, presumably because of the presence of ELL and EAF in this complex. And Ali Shalatafard's lab showed, and HEDA and now others have confirmed, that the little elongation complex is recruited to Pol2 transcribed snRNAs, which, as you know, are important for RNA splicing. And as I'll show you later in the talk, Amal Ranjan in our lab, working with Hide and his group in Japan, has now gotten evidence that the little elongation complex also regulates replication-dependent histone genes. So the identification of these ELL-EAF containing complexes as MED26 interacting partners really raised the question of whether MED26 via interaction, or whether Mediator, via interactions between the MED26 N-terminal domain and the EAF protein, might be able to recruit either the SEC or the LEC to promoters to be able to then influence the activity of genes of genes. And so to begin to address that question, Hide started by using a biochemical assay that takes advantage of the fact that Mediator can be recruited to immobilize promoter DNA, DNA immobilized on a magnetic bead through a DNA binding transcription factor. So the transcription factor binds to an appropriate sequence element on the DNA, recruits the mediator complex, including MED26, and Hide then wanted to ask whether Mediator could in turn recruit EAF and ELL and associated proteins. And indeed, what Hide saw in these experiments was that when these binding reactions contained both the mediator complex and an activator that could bind to the DNA template, both ELL and EAF were strongly recruited along with Mediator and the activator to this DNA template. But when the mediator was not present, EAF and ELL were not recruited. So that was nice, but he wanted to know whether a mediator that contained a mutant form of MED26 that lacked that N-terminal domain that interacts with the EAF protein could recruit the EAF protein to such a DNA. And our prediction, of course, was that it would not. And indeed, what Hide found was that while, again, the wild mediator containing wild-type MED26 would recruit elongation factors to this promoter in vitro, mediator lacking the N-terminal domain of MED26 could not. So these biochemical experiments then raised the obvious question whether the recruitment of ELL, EAF containing complexes to promoters by the MED26 N-terminal domain might contribute to gene regulation in cells. And so to begin to address the contribution of the MED26 N-terminal domain in cells, Hide wanted to generate additional mutants that would not be quite such a sledgehammer as simply deleting the entire N-terminal domain. And through a bunch of experiments that I don't have time to describe, he was eventually able to identify a number of point mutants, including these two, that would not disrupt the structure of the MED26 N-terminal domain or prevent it from interacting with mediator that could bind to RNA polymerase II, but that would completely obliterate the interaction of the mediator complex with these elongation factors. So Hide then went ahead to ask whether this MED26 mutant, R61A, K62A, could support MED26 function. Could support MED26 function in cells. And so to do so, he generated cell lines that express doxycycline-inducible small hairpin RNAs to knock down MED26 expression. And you can see he got a partial knockdown of MED26 expression and asked whether depletion of MED26 in these cells would cause cells to grow more slowly. And what you can see is that when he didn't induce the shRNA, the cells proliferated normally, but when he did induce the shRNA to knock down MED26 expression, the cells proliferated more slowly. He then did rescue experiments where he introduced into these cells either the MED26 point mutant that can't bind to elongation factors or wild-type MED26. And the controls where he's not introduced any rescue construct are shown in black here. And you can see that when he adds back the MED26 mutant that can't interact with elongation factors, there's no rescue of this proliferation defect. But when he reintroduces wild-type MED26, he now sees a big boost in the proliferation rate of these cells. So taken together, These observations then were consistent with the possibility that the recruitment of these ELL-EAF containing complexes might be required to promoters through MED26 or to genes through MED26 might be important for normal cell proliferation. Perhaps by altering the expression of oncogenes or other growth control genes regulated by the SEC, or by regulating the expression of snRNAs or histone genes regulated by the LEC, or both. So Hide went on then to ask whether, I keep hitting the wrong button, sorry. Hide went on then to ask whether MED26 was required for proper gene expression, and he initially focused on the oncogene c-MYC, which is regulated by the SECs, because in microarray experiments, he'd found that it's among the transcripts that's most affected, most reduced following MED26 knockdown, and it's a classic example of a gene that's subject to regulation during elongation. So now he's using the same knockdown cell line. He finds when he induces the shRNA to knock down MED26, he sees a reduction in MYC expression. When he puts back in the wild-type MED26 protein, he rescues that reduction, but when he puts back in the mutant MED26 that's not capable of interacting with the elongation factors, he does not rescue the MED26 dependent, or the MED26 dependent expression of MYC. Okay. So the next question then was whether these defects in cell proliferation and MYC expression in cells lacking wild-type MED26 could actually be due to alterations in SEC recruitment or changes in Pol2 elongation. So what Hide then did was go on to use chromatin immunoprecipitations to look at the consequences of knocking down MED26 expression at the c-MYC gene. And so he did chips using antibodies against either RNA polymerase II, total Pol2 or phosphorylated Pol2, or against SEC subunits, and analyzed DNA that was brought down in these chromatin immunoprecipitations by PCR, looking at regions in the promoter proximal region or marching further downstream in the c-MYC transcript. In the bar graphs that I'm going to be showing you, chips from control cells are indicated in black, and in the MED26 knockdown cells in orange. Okay, so first of all, Hide's data suggested that there was a relatively small effect of depleting MED26 on formation of pre-initiation complexes, because he saw that when he knocked down MED26, there was very little change in the amount of TF2D measured by TBP chip at promoters, and there was only a very small change or modest change in the amount of RNA polymerase II at the promoter region, presumably corresponding to promoter proximally paused Pol2 in these chips. But what he saw was that the amount of Pol2 as a fraction of control, the amount of Pol2 as you march further downstream in the gene was reduced to a greater extent. And more strikingly, he saw that MED26 knockdown led to a reduction in the amount of the SEC subunits, AFF4 or CDK9, all the way through the transcription unit, starting in the promoter region and moving further downstream. And most strikingly, he saw that knocking down MED26 led to really big drops in the amount of serine 2 phosphorylation on the CTD of RNA polymerase II, which, as I told you, is a hallmark of elongating phosphorylated Pol2, or of elongating RNA polymerase II. And so taken together, all of these data were consistent with the model that in addition to its well-established role in regulating initiation, Mediator, one more experiment to show you is simply the data that I just showed you didn't say anything about potential roles of the MED26 N-terminal domain in these processes. What Hide saw is that if he overexpressed a wild-type MED26, he would see big increases in the amount of serine-2 phosphorylation on the Pol2-CTD. But if he overexpressed the point mutant that can't bind to Pol2, or that can't bind to the elongation factors, there was no effect. Okay, so these data are consistent with the model that in addition to its well-established role in regulating initiation, Mediator through the N-terminal domain of MED26 can recruit SEC. How about the little elongation complex? Well, as I told you, LEC was known to regulate snRNA genes, and to address the potential contribution of MED26 to LEC recruitment, Hide Takahashi did chips of the little elongation complex subunit ICE1 at snRNA genes in either control cells or cells in which he knocked down MED26 using siRNAs. And in these pink peaks, you can see strong recruitment of the LEC subunit ICE1 to snRNA genes here, here, or here. But after he knocked down MED26 with siRNA, you can see in the black traces that the amount of LEC at these regions is substantially reduced. And so taken together, this data argues that not only does MED26 help recruit the SEC to at least some protein coding genes, it also recruits the LEC to snRNA genes. And as I'll show you in very recent experiments, we've collaborated with Hide in his lab to get evidence that MED26 also recruits the LEC to replication-dependent histone gene, or that it helps contribute to LEC function at replication-dependent histone genes. Well, at the point that this sort of published work was completed, a number of important questions relevant to MED26's function in SEC and LEC dependent processes remained. So first of all, does MED26 regulate SEC-dependent post-initiation events in transcription globally? Or is its function instead limited to a small subset of genes? And a reason that we suspected that could be the case was that quite a number of other mechanisms for SEC recruitment had been described. For example, Joaquin Espinosa and his colleagues actually showed or provided evidence that the kinase module, which as I told you, is rarely present in the same mediator complexes as MED26, could contribute to SEC recruitment at this subset of genes. There's also evidence that another mediator subunit, MED23, can play an important role in SEC recruitment, and a variety of other transcriptional regulators as well have been shown to contribute to this process. Now, a second question arises because the snRNA genes and the replication-dependent histone genes are really quite short. In fact, the snRNAs are only 100 or so bases long. And so one might expect that even a slowly transcribing RNA polymerase II would have no problem synthesizing these transcripts in a timely fashion. And so why would a complex containing ELL-EAF, the little elongation complex, where the only known function of ELL and EAF is to make Pol2 go faster, actually be needed for the proper regulation of these very short genes? And so to begin to address these questions, Amal Ranjan used ChIP-seq to take a global look at the consequences of MED26 depletion or mutation on Pol2 occupancy and CTD phosphorylation. And for these experiments, we've taken advantage of a CRISPR-generated MED26 hypomorphic cell line that was generated in the lab by Kai Chen. This cell line expresses a MED26 mutant that lacks the N-terminal domain of MED26, so it can't interact with elongation factors, but it can still assemble perfectly well into mediator that binds to Pol2. Unfortunately, it's only expressed at levels that are 20 to 30% of the wild type protein, so we can't rule out the possibility that some of the effects that we see are due to just a general reduction in MED26 level. Now, before I show you the data from Amal's experiments, for the benefit of any non-Pol2 aficionados in the audience, I just want to quickly remind you that at most protein coding genes, in a ChIP-seq experiment using total RNA polymerase II antibodies, one sees a strong peak of RNA polymerase near the 5' end corresponding to the promoter proximally paused polymerase, and lower levels of Pol2 across the gene body and extending downstream of the polyadenylation site corresponding to elongating Pol2. At or shortly after initiation, CTD repeats in the largest Pol2 subunit are phosphorylated at serine 5 by the TF2H kinase, and the serine 5 CTD phosphorylation signal pretty much follows the pattern that's seen for total RNA polymerase II, at least in mammalian cells. And concurrent with escape from the promoter proximal pause, as I told you, the PTFB kinase phosphorylates the CTD on serine 2, and what one sees in this case is a pattern of CTD phosphorylation that begins near the transcription start site, but gradually rises throughout the gene body. Okay, so Amal went ahead to do ChIPs with antibodies against total RNA polymerase II, RNA polymerase II phosphorylated on serine 5 of the CTD or on serine 2 of the CTD. And these are browser shots from his ChIP-seq data sets showing the MYC and the SNAIL2 genes. SNAIL2, like MYC, is among the transcripts that show the greatest decrease in expression after MED26 depletion. And it was among the small number of genes that he had looked at by qPCR-based assays, like the ones I showed you earlier. So in this thing, MYC is transcribed here from left to right, and SNAIL from right to left. Okay, so what you can see, focusing first on the total Pol2 chip, is that at MYC, the distribution of total RNA polymerase II in the wild-type cells and the mutant cells is fairly similar. In both cases, you see a strong peak of promoter proximally paused Pol2 and polymerase extending through the body of the gene, except that in the mutant, you can see that there's sort of an accumulation here in the 5' portion of the transcribed of the gene body, as if polymerase is piling up, and a decrease in the amount of polymerase, a minor decrease in the amount of polymerase at the 3' end of the transcript, as if the polymerases aren't all making it to the end. And you can see that phenomenon more dramatically over here in the case of the SNAIL2 gene, where there's a really substantial loss in total RNA polymerase II at the 3' end of the gene, consistent with the possibility that polymerase elongation is not working the way it's supposed to. So moving on to serine 5 phosphorylated CTD, in both wild-type and mutant cells, you see pretty much a pattern of phosphoserine 5 CTD that follows Pol2 distribution here and here. And finally, in serine 2 phosphorylated cells, we see quite dramatic differences consistent with what Hide had seen before in the PCR-based assays. You see that in the mutant cells, here at MYC and here at SNAIL2, there's a really dramatic decrease in the amount of total CTD phosphorylation. So at these two genes, MED26 mutation has a couple of effects. First of all, it leads to changes in Pol2 distribution that could be consistent with defects in productive elongation. And second, it leads to a really dramatic decrease, in serine 2 phosphorylation all the way across the gene body, consistent with the loss of SEC recruitment. Now, curiously, as we continue to scan through the browser, we noticed that this near complete loss of CTD phosphorylation across the entire gene body really wasn't particularly typical. In fact, what we saw at many, or I'd say even most genes, including these two, was that the decrease in serine 2 phosphorylation that's observed across most of the transcribed region of the gene here or here was accompanied by a really quite dramatic accumulation of CTD phosphorylation at the 5' ends of the transcripts here or here. And this accumulation of serine 2 phosphorylation extended over a region of about 2 or 3 kb from the transcription start site. Also noteworthy at these and a large number of other genes was that we saw some accumulation of Pol2 immediately downstream of the promoter proximal pause, or within the first couple of kb of the promoter proximal pause, in the same region as the area of increased CTD phosphorylation. And you can see that more clearly here. So there's a pileup, again, of RNA polymerase II within the first couple of kb of this transcript, overlapping the region with enhanced CTD phosphorylation. And one last point, these changes really are widespread. These metagene plots are showing the average distribution of total Pol2, phosphoserine 5 phosphorylated Pol2, or phosphoserine 2 phosphorylated Pol2. And you can see there are only really genome-wide, fairly minor changes in total Pol2 or phosphoserine 5 distribution. You can see a little decrease at the 3' end and this little pileup just downstream of the promoter proximal pause. But the phosphoserine 2 CTD again shows this really dramatic 3' to 5' shift in the serine 2 pattern. So what might this mean? And we really were scratching our heads over possible explanations for this result when this quite interesting paper from David Bentley's lab came out in Molecular Cell, in which he and his colleagues observed in human cell lines expressing slowly elongating Pol2 mutants, so mutants that transcribe much slower than wild-type Pol2, very similar phenotypes to what we had seen. So in particular, what they saw was that slow transcription evoked a hyperphosphorylation of CTD serine 2 residues at the 5' ends of genes. So they saw this same 3' to 5' shift in CTD serine 2 phosphorylation. And to explain that result, they proposed what they called a dwell time in the target zone model that they argued might explain the effects of transcription dynamics of elongation rate on the establishment of marks that are established along a gene body during transcription of the gene. And so what this model predicts is that if a modification reaction occurs on a time scale similar to the length of time polymerase dwells in a particular region as it's traversing the gene, then the extent of any modification that's linked to Pol2 in that region will be sensitive to elongation rate. So in this case, as the hypothesis would be is that as Pol2 is transcribing normally through the first couple kb of a nascent transcript, it's moving fast. The recruitment of SEC and the phosphorylation of the CTD is relatively slow compared to the length of time it takes Pol2 to move through. So there's relatively low levels of CTD phosphorylation, whereas in this case, the slow Pol2 mutants, polymerase moves slowly, which allows more time for a lot more serine 2 phosphorylation to take place in this region. And so in light of the similarity between the slow Pol2 mutant and the MED26 mutants, we think an attractive hypothesis is that because of a defect in Mediator's ability to recruit the superelongation complex, the ELL-EAF containing complexes to these genes, which we know will increase the rate of elongation by RNA polymerase II, this MED26 mutation might generally slow RNA polymerase II, perhaps throughout the entire bodies of genes. And so we're now in the midst of experiments that are seeking to address that hypothesis by directly measuring the kinetics of polymerase elongation in cells in these mutants. The other point that I should mention is that a little bit of a paradox. If the PTEF-B kinase is the kinase responsible for phosphorylation of serine 2 in the 5' end of the gene, and if, as I've told you, we think that the MED26 mutant interferes with SEC recruitment, which would mean PTEF-B recruitment, this set of observations might predict that additional mechanisms, perhaps some of those that I mentioned earlier in the talk, like recruitment of PTEF-B through the bromodomain containing BRD4, might be responsible for some of these early 5' phosphorylations of the CTD. And that, again, we're hoping to test in the fairly near future. Okay, so I've managed to run long. I'll try to go quickly now. For the rest of this time, I want to tell you about experiments addressing roles of MED26 in the regulation of genes that are occupied by and regulated by the little elongation complex. As I told you before, MED26 recruits the LEC to snRNA genes, and HEDA has evidence consistent with the possibility that it does the same at genes encoding replication-dependent histones. And as I mentioned earlier, it's not immediately obvious why you'd need these elongation factors at such short transcripts. And so that raises the possibility that perhaps the LEC might have functions other than just making Pol2 go faster. What these genes have in common, the snRNAs, is that they're unusual transcripts for Pol2 in that they're not polyadenylated. And as I'll show you, data from Amal Ranjan and Cynthia Chen at Stowers in collaboration with Hide Takahashi and his colleagues in Japan argue that MED26 and LEC regulate transcription termination and 3' end processing of non-polyadenylated Pol2 transcripts. So this story started with the observation that there were unexpected differences in how MED26 depletion or mutation might affect the abundance of certain transcripts, depending on whether RNA-seq was performed with poly-A-selected libraries to specifically measure polyadenylated transcripts or ribodepleted libraries, which measure both polyadenylated and non-polyadenylated transcripts. And in the heat maps I'm going to show you, red means the transcript went up upon mutation or depletion of MED26. Blue means it went down. And so we saw that when we used ribodepleted libraries to measure total RNA, the expression of a bunch of the replication-dependent histones went down modestly. But in contrast, when we analyzed the consequence of knocking down MED26 expression using poly-A-selected libraries, expression of the same group of genes appeared to go up, and in some cases, it went up a lot, tenfold or more. Now, a possible explanation for this observation is suggested by what's known about the synthesis and processing of the replication-dependent histones. So at the 3' end of the coding sequence of these genes is a stem loop structure in the RNA that signals processing of the nascent transcript into poly-A- transcripts and eventually signals transcription termination. If processing and termination fail, transcription continues past the stem loop structure, further downstream past a poly-A signal. And that leads to the formation of aberrant poly-A plus transcripts. Now, one explanation then for the RNA-seq results would be that processing and termination of these transcripts is inefficient in MED26 mutant cells, such that polymerase reads past the normal processing and termination sites, allowing formation of polyadenylated transcripts. And in fact, evidence from Amal's genomic data was completely consistent with that possibility. And I'll just skip over this point. But so in this top panel, I'm showing you a browser track for one of the replication-dependent histone genes of chips done with antibody against total Pol2. And the normal 3' end of the transcript is right around here, and what you can see is that there's considerably more Pol2 in this downstream region, as if the polymerase didn't terminate properly. Now the lower panel is showing you data from a so-called ProSeq experiment, which is a modified nuclear run-on assay that allows you to measure the position of transcriptionally engaged RNA polymerase along the chromosome. And again, you can see that there are a lot more reads downstream of the normal, the region where in wild-type cells transcription's sort of petering out up here. There are a lot more reads in the mutant cells, as if transcription has failed to terminate. Now, Hide Takahashi went on to use a PCR-based assay to look at the efficiency of processing of these replication-dependent histones. This assay measures the ratio of unprocessed transcript to processed transcript. So the higher the ratio, or the higher the value, the less efficient processing is. And so what you can see is that in the control cells, there's very little unprocessed transcript relative to total. And in cells in which he's knocked down MED26 using any of 3 different siRNAs, he sees a considerable increase in the amount of unprocessed transcript. He's also obtained evidence that the little elongation complex, like wild-type MED26, is needed for efficient processing of these replication-dependent histone genes. Again, he shows that knocking down MED26 leads to an increase in the amount of unprocessed transcript. The same is true when he knocks down ICE1, one of the little elongation complex subunits, but not when he knocks down AFF4, which you may remember is one of the subunits of the other ELL, like EAF-containing complex, the super elongation complex. So taken together, these observations could be consistent with the idea that the MED26 dependent LEC recruitment could be needed for efficient processing and termination of these transcripts. I mentioned that MED26 is also needed for recruitment of LEC to snRNA genes. And at these genes, another sequence, a so-called 3' box, provides the signal for processing into poly-A- transcripts and for transcription termination. And again, if processing and termination fail, transcription continues further downstream to cryptic polyadenylation signals in this case, leading to formation of aberrant polyadenylated small nuclear RNA transcripts. And Amal genomic data argues again that just as he'd seen in the histone genes, that MED26 is required for efficient termination at these LEC-occupied snRNA genes. So I'm going to just highlight the ProSeq data in this case. Here and here in the wild-type cells are the positions where RNA polymerase II normally terminates transcription. And you can see that in the mutant cells that lack the portion of MED26 required for interaction with the little elongation complex, you have a whole bunch more reads indicating that RNA polymerase II has continued past the normal termination site. So then, the question is, how might MED26 actually regulate transcription, termination, and 3' end processing of these replication-dependent histone and snRNA transcripts? Earlier findings had shown that 3' processing and termination of non-polyadenylated transcripts depends on a large number of proteins, only a few of which I'm showing in these rather oversimplified diagrams. And among these are something called the cap binding complex ARS2, or CBCA, the negative elongation factor NELF, which is needed for promoter proximal pausing as well, and RNA processing factors called CSTF, CPSF, and Simplecan in the case of the histone genes, the replication-dependent histones, and the integrator complex in the case of the snRNA genes. As I showed you, MED26 can recruit LEC to these genes, and Hide Takahashi and his colleagues in Japan have obtained evidence that the little elongation complex actually interacts with and helps to recruit CBCA, NELF, and the 3' processing factors to both the RDH genes, the replication-dependent histone genes, and the snRNAs. And so our working model at this point is that MED26 recruits LEC to snRNA and the replication-dependent histone genes, and that as a consequence of that, it's able to coordinate assembly of the 3' processing and termination machinery at these genes. So just to quickly wrap up, I've shown you evidence that MED26 can regulate gene expression globally by helping to recruit these ELL-EAF containing complexes, the SEC to protein coding genes, and the LEC to both protein and non-coding RNA genes that encode poly-A- transcripts. This is, to us, interesting enough, but we think there's also a potential implication of some of these findings for models for leukemogenesis by the MLL translocation partners that I mentioned earlier in the talk. And this is now going to be quite speculative, but testable hypothesis. So there's evidence, as I mentioned to you, that these MLL translocation partners that are normal components of the superelongation complex, when they form these MLL chimeric proteins, can be aberrantly recruited to particular regions in the chromatin. And one model that's reasonably popular in the field is that as a consequence of this aberrant recruitment, PTEF-B, EAF, and ELL, and other associated proteins are inappropriately recruited to promoter regions where they can be transferred to RNA polymerase II and allow inappropriate activation of elongation. But we think another sort of amusing possibility is that perhaps some of the oncogenic MLL chimeras could alter gene expression by causing inappropriate recruitment of mediator and RNA polymerase II, since inappropriate recruitment of SEC to particular regions in the chromosome is going to bring EAF to these regions. EAF can interact with MED26, which in turn could pull in mediator and RNA polymerase II to these genes. And so that's a hypothesis that remains to be tested, but we think one that warrants being addressed. And again, I'll thank my colleagues and I'll thank you for your attention.
Thank you for your nice talk. So I'm curious about the chip result of the PS2 POL2, which is the elongation form of the RNA polymerase II. So why is it the chip signal is so enriched in the polyadenylation site?
Why is the
chip signal so
of the
high?
serine 2 phosphorylated POL2?
Yeah.
Well, so that's been observed for a long time, and it's been proposed that the serine 2 phosphorylated CTD may contribute to the normal processes of termination downstream, which occur downstream of the cleavage and polyadenylation site in the transcription unit. So as polymerase moves along the template, reaches the polyadenylation site, the transcript is cleaved and polyadenylated, but POL2 just keeps going for quite a distance. And presumably you get then continuing CTD phosphorylation, perhaps as polymerase is slowing down again in that region,
so
preparing to terminate.
Okay, thank you.
So yeast have small nuclear RNAs, but they don't have MED26.
Right.
So how do they manage to process them?
By some other mechanism. You know, I don't want to argue that Med26 is that this mechanism that I've described is the sole contributor to proper processing. And in fact, I don't know if you noticed, but in those graphs showing the ratio of unprocessed to processed transcript in the Med26 mutant cells, it's still the majority of the transcripts are being properly processed. So this is not the mechanism for processing, but one of many mechanisms that's contributing to a proper assembly of the processing machinery. I do think it's striking, though, that the termination appears to be really much more inefficient than processing. If one looks at the distribution of proseq signal downstream of the snRNA genes or downstream of the histone, coding sequences, that proseq signal stays up for a long ways past the normal termination site. So it may be that the effect is much more dominant, or that the primary effect is on termination, and that in turn feeds back on the processing. I don't know.
Thank you.
It's a beautiful talk, and a naive question. So what's the consequence with this processed and unprocessed transcript in terms of this RDH gene?
Yeah. So I'm glad you asked that question, because I sort of I blew right past that point in the interest of time. So there's a really interesting paper that came out from Bill Marsliff's lab in the last year or two, showing that, well, first of all, I should say it's been known for this business that you can have inefficient processing and termination of the histone genes, ultimately giving rise to polyadenylated transcripts, has been known for a long time. And some of the histone genes are actually made as either replication dependent, cell cycle dependent, non-polyadenylated transcripts, as well as constitutively expressed polyadenylated transcripts. And the H2AX is one notable example of that. But what Marsliff's group showed was that there's a population of genes that are normally expressed in proliferating cells, meaning the ones we all study, HeLa cells and stuff. They're normally expressed as cycling transcripts, and they've been considered to be sort of strictly replication-dependent histones. But then that begs the question of, how do you maintain your histone population in very long-lived, non-dividing cells, like neurons that can last for a really long time? And so they went in and asked whether there might be a population of what we think of as replication-dependent histone transcripts that in these non-dividing cells are made constitutively as polyadenylated messages that are more stable and better able to. And they found that indeed quite a number of the replication-dependent histones are made as polyadenylated transcripts in neuronal cells. And even some of them are spliced. So in order to access a polyadenylation signal, they remove an intron in the 3' UTR region that allows them access to a distant poly-A site. So, a sort of amusing possibility is that as one means of contributing to that sort of switch from non-polyadenylated to polyadenylated transcripts, if Med26 is less abundant in non-proliferating cells, which is in fact the case, you might imagine that decrease in this mediator function could potentially contribute to that regulatory switch. But that's, again, wild speculation at this point. Yeah.
I have a question on the Pol2 CTD heptide repeat phosphorylation aspect. Presumably, in each of the transcriptional machinery, there's only 1 or 2 copies of each of the subunit, each of the protein, right?
Say again. So in each
During each Pol2 machinery transcribing DNA into messenger, so only one overall Pol2 machinery working on 1 copy of DNA, right? So you also alluded to that the moving speed might determine the dwelling time of the kinases phosphorylating these repeats. So what is the mechanism counting several dozens of the repeats of phosphorylations? Exactly what might be going on?
I'm smiling because the expert an expert in that field is right behind you. So one thing that's clear is that, at least in the test tube, the protein kinases that phosphorylate the carboxy terminal domain are quite processive. So if you add kinase to Pol2-CTD, rather than seeing, in the case of the TF2H kinase that puts the serine 5 phosphorylations on, as well as the PTFB that puts the serine 2 phosphorylations on, you don't see sort of a uniform gradual shift in electrophoretic mobility as the various repeats are phosphorylated. Instead, you see a sudden jump. So the first thing you see is that a population of the enzyme shifts up, while most of it is still running at the electrophoretic mobility of hypophosphorylated Pol2. So it may be that a single kinase can, in principle, put a lot of phosphorylations on one CTD. And I'm not sure if that answered your question, but...
So after certain amount of phosphate being added onto certain repeats, who reads it, decodes that information?
Who will decode that information? So the CTD is thought to act as a landing pad for other enzymes that also mediate various co-transcriptional processes. So, for instance, the serine 5 phosphorylated CTD at the 5' end of the gene provides a landing pad for the enzyme that catalyzes capping of the messenger RNA. The phosphorylated CTD is thought to help to coordinate assembly, or it's hypothesized to coordinate assembly of splicing machinery for co-transcriptional splicing. There are chromatin modifying enzymes that interact with phosphorylated CTD. So serine 2 phosphorylated CTD recruits a histone methyltransferase, for example, that places H3K36 methylation on the nucleosome. So one possibility is that it's providing sort of a cloud of binding sites around the enzyme that then help to bring in other stuff that needs to do things during transcription. Okay.
Can I ask a question?
Sure.
So you show that in your MET26 depleted cells, that c-MYC and snail are rather the exceptional case, right, against 9,000 other genes.
Yeah, I mean, I
So-
wouldn't say that they're the only case.
Sure.
But-
So I was just curious if are there any kind of features in c-MYC
Yeah.
and snail in terms of like genome, like packing, or other features based on sequence that you can tell that makes them different?
We haven't figured out what makes them different. They're both short. I don't know whether that has anything to do with it. They're both, as I said, among the genes that are most downregulated upon MED26 depletion. That said, this more common shift of the serine 2 mark from 3' to 5' is seen in genes that are downregulated, in genes that are upregulated, in genes whose expression doesn't notably change. So, no, I don't have a clue why those appear to be different. And really, the only reason for highlighting them was to make the point that these cells behave the same as what we'd seen in the earlier assays. But it's a really, really interesting question why those ones are behaving differently, and we just don't know yet.
Thank you. Oh, one more. Maybe this is the last question in the interest of time.
Sorry, I have a question about EAF1 and EAF2 identified as ELL associated factor.
Yes.
In that slide, I saw there is, beside from the full length, I saw a lot of lower band that have similar pattern, but after you add the EAF1,
Yeah.
the pattern start to change, and I was wondering, is those pattern are due to pausing or what other reason? Because like, although usually we care for the full length, but why? What is the reason that the transcript stop at multiple sites and they have a
Right.
uniform? Although the expression is not the same level, but it's have a pattern that we can observe.
Yeah. Let me see if I can rephrase your question. So are you asking, for example, you might imagine intuitively that if you start with a short transcript and you watch polymerase move along the gene, that you'd see a fairly uniform distribution of products getting longer and longer with time. But instead, what we see is this wide distribution of products, some of which stay relatively short and some of which are actually pretty long. Is that, and you're wondering why that might be and
Yeah,
what
because
what EAF might be doing?
like if like they have a gradual trend that they stay at the top and they increase gradually to
Yeah.
the top, then I think it's reasonable. But I saw some of the band, they are more intense in the middle.
Yeah, and those sites would correspond to places where polymerase more often than not might tend to pause. And I think in that EAF assay, I only showed you a single time point. So you're catching a snapshot where most of the polymerases haven't made it all that far along the transcription, along the template. And so polymerases are tending to pause at different places. So why don't they all pause at the same place then? So it was observed a long time ago that polymerases, while they're elongating, at sort of each step of nucleotide addition, will stochastically either add a nucleotide really fast or sit there for a little while and before they add the next nucleotide, presumably because, now we have structural data from Dong and from many other people showing that the polymerase has to be in the right conformation with the nucleotide lined up just so in the active site with the 3' end of the nascent transcript in order to add a nucleotide quickly. And so very quickly what happens as polymerases are moving up the template is that some of them happen to go fast on average, some of them happen to go slower on average. But there are particular sequences at which polymerase tends to have a harder time than others in adding the next nucleotide. And at those places, then you tend to see polymerase pausing and a band appearing. And that's accentuated in the particular assays that I showed you because we don't have fancy kinetic equipment in the lab. We're relying on our hands to be able to pipette and to stop the reaction at different times. And so to make that experiment easier to do, we usually use quite low concentrations of nucleotides And sometimes one of the nucleotides we have is out of balance and is actually at a really low concentration. And that means that every time the enzyme hits, for example, a place where it needs to put a U into the transcript, it might tend to slow down, and then you might see a banding pattern.
So is it an intrinsic property of the isotope based?
It's not an intrinsic property. Well, it can be an intrinsic property of the sequence, and the pausing pattern can be sort of superimposed on the sequence. You can cause the pattern of pausing to be a little bit different. For example, depending on whether you use a low concentration of ATP or a low concentration of CTP. In those elongation assays, the low concentration is not because of the isotopes that we're using, because we actually dilute the isotope so that we only see the elongating product. We can talk about it more afterwards if you'd like to.
All right, thanks very much, Joan, for
Thank
a
you.
wonderful lecture. And
And thanks to all of you.